This protocol is significant because it allows for an in vitro analysis of neutrophil swarming, which overcomes many limitations that are present in an in vivo experiment.
The main advantage of this technique is that it provides easy access to the secreted cytokines and lipid mediators that neutrophils release during swarming and allows for quantitative imaging analysis. While we applied this technique to neutrophils, it can be extended to analyze the migration of other white blood cells like monocytes and T cells.
Begin by preparing a 1.6 milligram per milliliter cationic polyelectrolyte, or CP solution, by adding the proper amount of CP powder to water and mixing it on a stir plate overnight or until all solids are dissolved. If desired, the solution can be made fluorescent by adding polyl lysine labeled with VitC.
Generate the master silicon wafer using standard photolithography procedures.
The design used here is a 4x4 mm rectangular array of 30 micrometer diameter filled-in circles with a 150 micrometer center-to-center spacing, but it can be modified as desired for different applications. Spin coat a 40 micrometer thick layer of negative photoresist onto a silicon wafer. Then bake the wafer at 65 degrees Celsius for 5 minutes. And 95 degrees Celsius for 10 minutes. Expose the wafer to UV light through a chrome photomask with 150 to 160 millijoules per centimeter squared.